Welcome to Data in Biotech, a podcast from Core9 where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Today, we're joined by Dipen Sangurdekar, VP of Data Sciences at KSQ Therapeutics. Dipen takes us into the world of cancer therapy, focusing on tumor-infiltrating lymphocytes and the groundbreaking applications of CRISPR-omics technology. He and Ross discuss the challenges of translating preclinical discoveries into clinical success, the complexities of drug development, and the IND and clinical trial process. Dipen also shares why integrating data science with biology is crucial for driving innovation in cell and gene therapy. Here we go.
Dipen Sangurdekar, welcome to the Data in Biotech podcast.
Hi, Ross. Good to see you.
Awesome. So just to kick us off, could you give us a brief introduction to your background and what brought you here today?
Absolutely. So my name is Dipen Sangurdekar. I'm currently a Vice President, Head of Data Sciences at KSQ Therapeutics. KSQ Therapeutics is a oncology biotech company based in Lexington, Massachusetts. Our mission is to solve or cure solid tumors with engineered cell therapies and one and done sort of cell therapies. If you're familiar with cell therapies, they can be broadly of two kinds. One is autologous, where we take patients' own cells, engineer them with either CARs or TCRs, and put them back in. And the other broad category is allogenic, where you take cells from healthy donors, potentially do some engineering to make sure that they evade host immune attack and put them back in after equipping them with CAR and TCR. So our cell therapies are called TIL therapies, which come in the autologous category, where we do all the engineering. However, instead of attaching a single CAR or a single TCR, we actually take the cells from patients' tumors. These T cells have been trained by their own body itself to attack and fight those tumors. It's just that they have gone through the process of attacking tumors and gone down the exhaustion path and perhaps need a little boost. So we take those cells out. They are called tumor-infiltrating lymphocytes, or TILs for short. We take them out, we expand them, recharge them, and infuse them back. Our sort of differentiation there is that using a platform called CRISPR-omics, which is genome CRISPR screens, we have found gene targets, which we believe by knocking them out, really turbocharge these cells and help them overcome the difficult environment that these cells typically see, especially in solid tumors. So overcoming immunosuppressive environment, cytokine shortage, and so on. So we are using the CRISPR technology both as a discovery tool, but as a therapeutic tool as well to modify our cells.
Awesome. Well, that's a fantastic introduction to KSQ Therapeutics and to TILs, to tumor-infiltrating lymphocytes, and to your background. Before we jump into sort of the fun data science type work that you're doing at KSQ, can you just tell us, why did you get into this field in the first place? What led you down this path?
Like I said, I think the first day of my graduate school, I picked up a pipette and started doing genomics experiments, which included doing genetic screens and a lot of transcriptional and microarray work. And having come from a quantitative engineering background, those early days in the lab sort of really opened up the complex world of biology, right? Fundamental research, life sciences, and also what it could mean for, sort of health, understanding more complex systems such as human systems and disease. So I really wanted to be on that interface of sort of generating those hypotheses in a data-driven manner and having the ability to query those hypotheses in the lab and doing that sort of cycle time, which back then, maybe it was not as an obvious of a trajectory, but now, of course, you have an entire generation of scientists that are cross-trained between data science and experimental techniques. So yeah, I think it was a matter of continuing looking for interesting problems to solve using my toolkit that I was slowly developing over the years and understanding of the platform, the genomic technologies. And one thing led to another, and that's where I am. Over the last few years, focused on the interface of how preclinical work or biology, what we call preclinical, means when we use model systems, either in vitro cell lines or in vivo data, how that understanding of mechanism of action of our drug targets translates into the clinic, and how we can learn from clinical data, because we generate a ton of data, even from a quote unquote failed clinical trial. We can generate a lot of data that we can take back to the clinic, either to diagnose or to improve drugs. And being on that interface is just, I find it very fulfilling and thrilling, and something that I feel I can contribute in a very tangible way.
Yeah, that's awesome. And I really want to return to that question of, how preclinical work and clinical work create feedback loops between each other and how you translate between the two. But before we do that, I want to return to TILS and the, sort of the data-driven work you're doing there. So you mentioned the CRISPR omics platform, and that it has sort of like two applications that it's used for. There's the sort of the target identification aspect of it, and then there's the therapeutic intervention aspect of it. Can you just walk us through, maybe just a brief introduction to CRISPR-Cas9 and, what it is, and then how it's used in these different contexts?
So, as perhaps many of your audience already know, the CRISPR-Cas9 system, which has won Nobel Prizes in the field, is used to systematically create double-strand breaks in a mammalian system, right? So, let's say a human cell, you want to precisely at a particular genomic coordinate go in and create a double-stranded break. Because of the DNA repair mechanisms, those double-stranded breaks often get repaired in a way that causes what we call a short insertion or deletion. And if done right, if you find the right location in the gene, that can knock out protein function for that gene. So, genetic screens, knockout screens have existed prior to CRISPR, but CRISPR has really enabled doing this at scale, especially in complex mammalian systems and human systems. So what we have done and others have done as well is use libraries of these synthetic guide RNAs, which is what directs the CRISPR-Cas9 enzymes to particular location in the genome to cause those edits. So you can multiplex those, you can create libraries of those, and there are publicly available libraries, as well as, it's fairly straightforward these days to create libraries computationally. You can multiplex those and create screens where you take cells of interest, right? Whether it's a cancer cell line, whether it's an immune cell, either in an in vitro setting, such as in a dish or in a mouse model, and you conduct an experiment, what we call a forward genetic screen. So essentially, you're letting a bag of cells, each of which hopefully has one edit, right? And you can control your experimental parameters to make sure it's only one edit per cell. You let them compete, right? Whether what we're looking for is a functional readout, such as an expression of a surface protein, or if you're looking for how well those cells kill a particular cancer cell line, or how well those cells migrate to a particular location in a tissue. Once you do those experiments, you can sort either using flow cytometry, or you can do extraction of a tissue, and you can do NGS to find out which sgRNAs are enriched. And that's a proxy for which edits are winning in this sort of almost like a survival of the fittest kind of situation. And then once you figure out which genes are knocked out, then you can, obviously that's not, you can't just take the top gene and then take it to a target validation. You have to then layer in all of the biology and all the prior knowledge that we know about those genes. You can do pathway analysis, you can compare it to other screen data. And you have to be very judicious in sort of down selecting which of those genes we want to take to a target validation funnel. And, that's how, once it's in the funnel, that's, your standard drug discovery, right? Then you can either design a therapeutic modality, which is such as a small molecule to target those genes, or a biologic, or in our case, design an ex vivo CRISPR editing system as a therapeutic by itself.
My understanding of this is that, basically this CRISPR omics-based system allows you to look at the entire genome and sort of go gene by gene and do like turn on and off decisions at the cell level, and then just sort of see which cells perform in the way that you want them, to perform, or demonstrate the characteristics that you want them to generate, or don't generate the characteristics that you don't want them to generate, that you can identify the types of genetic modifications that you could make therapeutically that lead, the cells that you're taking out of people and putting back in people to perform inside of the tumor in the way that you want them to perform inside of the tumor. Am I thinking about that right?
That's absolutely right. And in the case of you sort of mentioned one application, which is, we want to potentiate the cells in our case, right? So that they can go, they have increased functionality, and whatever that functionality is, you can define it in multiple ways, or whatever attributes you want to design into the system, and they go in and do a job better than if they were not edited, right? But the other way is you can actually just find targets. You can do those screens in cancer cell lines, which is something that we have also done, and find targets in cancer cells that under the right context kill the cells, right? So we have identified one such target, USP1, which comes into the sort of the class of synthetic lethality targets, because in the right genetic context of homologous recombination deficiency, knocking out USP1 or inhibiting USP1 leads to cell death, right? So since the discovery at KSQ Therapeutics developed a first-in-class small molecule inhibitor called KSQ 479, and now that program is being led by Roche further into clinical development. The discovery can sort of downstream be taken into a variety of applications, and that's where the therapeutic drug discovery part comes in. And in the context of our gene-edited TILs, they're basically using the CRISPR modality itself, where we design a therapeutic grade synthetic guide to create those gene edits in patients' sort of TILs themselves, right? Ex vivo means outside the body at a very high level of quality. So since this is a drug, we are held to a much higher standard of quality. And when I say quality, in this case, we want to make sure that we are editing where we say that or where we intend to edit, and we are not editing elsewhere in the genome. And and we are editing to a very high level, right? So it's not like we are only achieving 5% of the cells being edited. We want to have a very high fraction of cells to be edited for it to be a therapeutic. So that's where it comes into the the CMC, right? That this suddenly becomes a manufacturing thing that we have to pay a lot of attention to in terms of design of the therapeutic guide, the controlling the parameters of doing the CRISPR-Cas intervention for each and every patient that we treat.
Yeah, that makes sense. And so, I mean, as I'm hearing you describe this process, what I'm realizing is that, we use this term personalized medicine, but there's sort of multiple levels to the personalization here. There's sort of the ways that the genetic modifications that you make will have sort of dependent outcomes for the patient, depending on the nature of the rest of their genome, the nature of their biology, the nature of the way that their immune system is responding to the cancer, and the nature of the cancer itself. And then there's also the personalized nature of the medicine, because you're doing an autologous therapy where you're taking the patient's own cells and then modifying their own cells and then, sending them back to them to get them reinfused. And am I thinking about that right?
Yeah, yes. So there are two things here that we have to think about, right? And sort of this is sort of not talking about our product, but in general, whenever you sort of go down that discovery process, the edits that you find, right, or the targets that you think are supported by the screen or they fall out from the screen, you have to repeat those experiments in a variety of different contexts, right? Whether if you're doing this in mouse models, you have to make sure that you're testing this in mouse models with different genetic backgrounds. If you're doing them in cancer cell lines, you want to make sure that you're doing the screen in multiple cancer cell lines, because you can't just say that in a particular model you see a target being prioritized. That target is necessarily going to be the same, equally efficacious in other genetic backgrounds, whether it's in human cell lines or in mouse models. So, us and others who have done this, right, they ensure that this target is a valid target in a variety of those different contexts. And this is before we think about the translation from preclinical to clinical, because in the real world, there are multiple effects, right, that come in, right? Obviously, everyone has a different genetic background. And so this is where we want to make sure that our drug will be effective for everyone who has cancer and not just a particular subpopulation. They have their own sort of medical history. Typically, these therapies come in later in sequencing. So they have gone through multiple rounds of treatments, which can affect the quality of their immune cells, but also the tumors are much more refractory. They have a lot of genomic enrichment done. Their tumor microenvironment could be very immunosuppressive. So there are a lot of variables that come into play even in the clinic. And that's why the translational hurdle from preclinical to clinical, one of the things I really wish for is that using all the data, analytics, and AI, we could make that hurdle more of a gap rather than the chasm that it is right now. Because if we can make predictions preclinically that will translate into the clinic, even if we improve our success rate by 10 to 20%, that will be a significant benefit. It will reduce drug development cost, it will reduce failure rates, it will benefit patients, it will be a win. But that's where the gap is.
Yeah, I was going to wait to go there, but let's just go let's just go there now. So what does that look like? How do you think about improving your ability to translate from the preclinical work that you're doing in, finding the target and validating the drug to the clinic? And then how do you use the data that's coming out of the clinic to learn from the clinical data to improve the drugs that you're developing?
Yeah, I think one thing I already touched on that, making sure that any sort of funnel that you're constructing for target discovery includes looking at multiple model systems with genetic backgrounds. So that still does not reconstitute human biology per se, but at least you're assured that this is not just a artifact of looking in one cell line or one mouse model, that you need to have multiple ones. The second piece, which is definitely the harder thing to do, is trying to reconstitute what the human physiology looks like. In the case of oncology, for example, trying to recapitulate tumor microenvironment in model systems, and people are making progress with that with organoid systems that try to mimic what the immune system, the microenvironment looks like. And there are multiple cells involved that are pro or anti-tumorigenic or pro and anti-inflammatory. So yes, we are making progress, but that's the key, right? A model is basically, by definition, something that captures, right, all the parameters that matter. And that's going to be key, right? I think these systems are coming online. People have been using them to make predictions. I think leveraging them, understanding them, generating single cell data and multi-omics data, and then characterizing what happens will go a long way in de-risking the translation across that gap.
Yeah, no, it strikes me that, your point about the clinic is where your model of the world meets the real world. And so the clinical data is teaching you what the gaps are between the real world and your model. And so the feedback loop looks like identifying the questions you didn't ask when you were building the model and the parameters that you didn't know when you were building the model, so that you can sort of iteratively refine that model based on the gaps in your understanding that led to unexpected effects once the therapeutic got into the world.
And I think you're hit on a very important point, right? We don't know what to build into the preclinical models if we don't study what the clinical data is telling us. And for that, we need to have a sort of an expansive approach, obviously within the budgetary constraints of running a clinical study, but we need to understand at a very detailed level once our drug goes into a patient. And remember, like, patients are really doing us a huge favor by participating in those studies, because honestly, a lot of the time these studies fail, right? But what we learn and what we owe to the patients is leveraging all the data that we can, so that in the future, we have a better shot on goal. And so in this example, I've been part of programs, not just here at my current company, but previously as well, where we have derived not just the clinical data captured in CRFs and clinical databases, but also patient sample data, right, with consent, right, to do exploratory research. So we often extract tissue, like cancer tissue data from biopsies, serum, that is blood, radiological imaging data, or other imaging data. These are non-invasive techniques that are part of the standard of care. So it's not like we are asking patients to do anything out of the ordinary. But as part of the standard of care, these samples are collected. We then generate multimodal data sets from those. And really, that's when we start to learn, right? Even if a drug did not work in the sense that, it did not produce a top-line benefit, can we see something in the in the PK/PD data? Can we see some pharmacodynamics? Can we see some sort of target engagement? And then we correlate, or can we build machine learning models that include the other data that we are collecting that explains that outcome? Because that is obviously, correlation is not causation, but then when we sort of know what we know about the mechanism of action, what we know about prior biology, we can really come up with new hypotheses that we take back across the hurdles to the bench, build them into a model system, tweak, and do the loop as you mentioned.
Interesting. Yeah. So I love where we left off, because it brings me back to, where I was looking to go earlier, which is, so if we can zoom out a little bit, that's an excellent example of where the data science team that you oversee, engages in connecting what you're seeing in the clinic back into, what you're doing to develop the therapeutics in the first place. Can you just walk us through broadly how the data science team, the combination of computational people, of bioinformatics people, and machine learning or AI people sort of work together to support the creation and then the optimization of the cell-based therapies that you develop?
So, if you look at it from a very high, sort of 30,000 foot level, right? I think the mental model that most people use, right, it's still very valid, right? Which is you have a pipeline, right? And you have an early stage of the pipeline where you have target identification, target validation. Once a target goes through the stage gate of becoming like a development candidate, the next sort of part of the pipeline is IND enabling studies, where we IND is investigational new drug, which is basically an application to the agency or FDA to say that, hey, we want to go into the clinic with this drug, and here's all the safety and efficacy data and some other background about our drug, right? So those studies need to be done, they need to be submitted, and then once a drug gets into the clinic, then we have the phase 1, phase 2, phase 3 clinical study. So it's still in that linear model is quite representative of how we think about it. So we have folks that, and again, I'm sort of generalizing what our team does, but also my previous teams and other teams that I've observed. There are people who sort of either are focused on particular stages of the pipeline for what they do. For example, someone is very focused on discovery, which is target ID, and often that's associated with some sort of a platform development, whether that's an AI platform, whether that's a screening platform. So focusing on that phase. Others who either travel with the project down that pipeline or focus on translational work are really skilled in understanding not just preclinical biology, but also are well versed in the data science aspects of what clinical data looks like, what are the different formats, what are the constraints and quality issues, and very importantly, how do you treat clinical data with care? Obviously, apart from HIPAA concerns, we have to make sure that the data is handled with appropriate integrity, security, and governance. Folks who are well versed in that can operate with the interface very effectively. I don't want to call background, because I think having a learning mindset is more important. People who are willing to learn, looking backwards, more about preclinical biology, what do model systems look like, what does the data really mean, and also what the clinical data looks like, what are the different terminologies, how do clinical sciences and development and translational research thinks about those problems. Folks who are willing to learn those two and bridge or make the bridge and use data as that connective tissue or data systems, those will thrive at that interface. And then there are, obviously an essential part of this that I want to make sure that I'm covering is, you know, there is the data engineering, right? And the technologies aspect of it, right? So we want to make sure that across the pipeline, we have the right systems, right? Technology stacks to make sure that the data is ingested, it is curated, it is inventoried, it is tagged, and it is housed in a way such that people who need to access the data have immediate access to it, whether you're using a scientific data management system or a LEM system, whether you're building bespoke databases, whether you're using cloud technologies and AWS services to access it. We need to have a strategy in place, and there are folks who own that piece as well. So we consider that platform work, foundational work, which supports everything, but no less important than any particular project. So I hope I answered that question.
No, I think you did. You've given it to us at a high level. Are you able to share any examples of like what a project looks like in the target ID or platform development part of it, or in the translational work, and what data you bring together, what methodologies you apply, that sort of thing?
Yeah, so maybe I'll give sort of a couple of examples, right? So, in one of my previous roles when I led a computational biology team, one example was they were working on a particular hypothesis to find. So the hypothesis was we want to go in and find targets for a particular cell type, right? And that target can translate. If you have sufficient hypothesis of a target, then we can modulate it. We can modify the behavior of the cell type. Now, we didn't really necessarily have any internal data on that cell type at the single cell level, so we ramped up a campaign of sourcing in tissues and doing analysis, data generation on the cell types, whether it's IHC data, single cell RNA-seq data, and so on. I think members of my team at that time focused on bringing in large public data sets. And this includes, but is not limited to, PCGA data and other single cell data sets that were published. So we built a large catalog of single cell data sets that existed out there, and we had brought in-house. We worked with a couple of companies in the space that did data curation and inventorying for us. And once we have the data, then we had a very specific question: what are the targets that are expressed in that particular cell type that are unique to that cell type and are not expressed elsewhere. So we want to, that's the one of the thing, right? There has to be a therapeutic, for there to be a therapeutic index, that target has to be expressed, preferably on the surface, right, in that target cell type, but not elsewhere. And that sort of leads you down and bioinformatics. That's a very sort of linear concept to follow and work through, right? And then once we have a list of targets, when you bring in other data modalities like IHC data, literature, obviously the huge amount of knowledge that scientists themselves carry from their training, and then it sort of all comes together. The other example I would give is sort of the screening data, right, that we do in-house, which is exclusively in-house data. That is our bread and butter in the sense that we generate large screens. And essentially, we modify, we sort of use libraries that we have synthesized to knock out genes at scale, but one gene per cell. And essentially, we do these enrichment studies where we are by sequencing methods, we are at the end of the, at a particular endpoint, we are seeing which of those sgRNAs are enriched or depleted. And, that's again a very linear concept, but it has a lot of nuance. So you have someone who is leading that analysis project, for example, would have a very deep understanding of not just what the experimental design is, but also informing the experimental design by saying, what's the number of replicates do we need? What kind of sequencing depth do we need? What's the power do we need, right? And do a lot of simulations to set up the experimental parameters, but then also have the skill set, bioinformatics skill set to do the next generation sequence analysis, do a very high level of quality control. And then finally, at the end of the pipeline, when the data comes out, then we have to come up with the statistical methods, rigorous methods to prioritize and come up with, the volcano plots and the lists, the rank lists that then go into downstream interpretation. So yeah, depending on the project, we could use internal data, we could use external data. Even for external data, it's not like it's clean, right? External data comes with a lot of metadata. Anyone who has downloaded data from Geo before knows, right? There's a lot of work that needs to go in to curate, clean up data, to do normalization, to merge the data together. So there's, it's not just that we start with a clean table and apply statistical models. A lot of thought goes into getting the data into a shape that can be analyzed downstream.
Yeah. So, what I'm hearing is that it's highly hypothesis driven, and that, the data sets that you bring together and the sort of methodologies that you apply are very much dependent on the hypothesis that you're chasing down. So if I understand that correctly, how do you keep track of all the hypotheses that you could be chasing down and sort of prioritize the work of the data science organization to make sure that you're, sort of driving the insights that the organization needs to move along the most strategically important?
And that's a great question. I've been fortunate enough to work in organizations or departments where data science teams or computational biology teams or bioinformatics teams that I've led have been highly integrated and embedded with the biology teams, whether they're discovery or translational research and so on. So to answer your question, we don't do this in isolation. Every member of the team is highly integrated into project teams, right? And the project teams are the one that come up with the questions, the hypothesis, right? And then if you're a data science member, part of that team, your job is not to just to react and do the analysis, but to bring your own perspective within the data sciences team or bioinformatics team. What we try to do is make sure that everyone knows what other people are working on in their projects, because that's where a lot of the cross-pollination of the data-driven hypothesis happens. That individual member can go back to the project team and say, hey, someone else is working on their project, and this is the idea that they applied or data-driven hypothesis that they generated. Maybe we could try a variant of that here. It's important not just to be reactive to the questions that are generated by the biology team, but also to bring unique ideas, innovative. That's how you grow as a scientist, as an expert. And that sort of raises all tides, I would say, across the board.
That makes a lot of sense. And also, bringing your expertise means also raising your hand if you believe that the questions that are being asked are not the right questions, that if the hypotheses they're being selected to work on are maybe not the most important hypotheses. But to your point, that's something that needs to be decided as an entire projects team for moving the project forward, rather than something that you sort of unilaterally do in the data science org. I'm interested in how working on cell therapies, on, these living drugs, makes these sort of machine learning oriented or statistical analysis oriented data projects hard. So what are the biggest challenges that you face in that environment?
Yeah, and that's another great question. I think one of the reasons why I really, I've been working in the cell therapy space, the gene and cell therapy space for a few years now. And one of the reasons I'm excited and I feel like as a data science practitioner, I have, something to contribute here, perhaps other than, more than other modalities is because, as you said, our drug product is highly personalized and it's a living drug. And what that means is that unlike a small molecular biologic, there are a few axes or dimensions in which you're optimizing the drug and defining a target product profile, right? Whether it's chemical matter, property, ADME properties, and so on. In a living drug, there could be many different dimensions, and we may not even know what those dimensions should be or are when we start to study. As an example, right? If in the field of CAR T, right, I think it's been well established that you need healthy cells, proliferating cells that also have the capability of going to the target, doing the effector function, and then living for a long time in the system. So initial efficacy, but also duration and persistence of those cells is important. Now, if you just look at those dimensions, double click on those, that could mean many different things at the biological level. And the only way to interrogate that is to do very deep profiling of our drug products before they go into the patients. Often, if you can, and if you can find them once they're in the patients, extracting them either from tumor biopsies or from the blood, if you have ways to pull them out and do characterization there, that's important because then what are the properties that you built into the system. And just like, what does the body do to the cells, right? That's also important, right? So you have a plan, and then when the plan meets the real world, what happens to that plan? Understanding both is important. And the only way we can do that is very deep molecular profiling using multimodal analysis, single cell, RNA-seq, in our case, TCR sequencing, but also proteomics, cytosis, etc. Now you have a large data cube, right? Very complex cube. Now, unfortunately, like when we talk about AI, we talk about scaling laws, right? We're not talking about a lot of scale. We are talking about a lot of depth, but not a lot of samples, just because of the space that we are in. So then we have to be very creative and asking those questions where you can't just throw machine learning models at it or build foundational models and say that, okay, patterns will emerge. We have to ask some very guided questions because we don't have enough data to escape the multiple testing thing. If you just do a fishing experiment, you'll get thousands of p-values and some of them will be significant just by chance. But if you go in with guided questions that are driven by prior hypothesis, you might get a signal that is weak, but suggestive of a trend, backed by a very strong hypothesis that gives you a lot more confidence that, hey, maybe this is real. And now let's go back and test this preclinically or non-clinically. And that's how that cycle works, right? Sometimes over time, you build a large enough data set and then you can do exploratory data analysis. You can then see if broader patterns emerge that you are not looking for and that correlate with a particular trait or a downstream outcome. That can happen, but in the early days when the data is just evolving, you're nowhere close to that. So you have to be a bit more smarter about what kind of questions you ask.
So, rather than letting the data tell you the story, you go in with the hypotheses that you believe might explain what you're seeing, for example, the difference between the cells that you've edited and deeply profiled before they go into the patient, and then the ones that you've deeply profiled after they go into the patient. You use your hypothesis to shine a light between them and then see whether there's any degree of signal there. If there's enough signal to support the hypothesis, even if it's not rejecting the null hypothesis, you can still move on to some sort of experimental validation that is also a more targeted approach and a more rigorous approach, but obviously more resource intensive. So you have to,
That's absolutely the right. Yeah. Thank you for, that's absolutely the right thing to do. I mean, again, there are statistical purists out there who might say, this is like, you're doing things. Experimental validation, the ability to go in and to quickly validate something frees us from those constraints of, getting a very strong a strong signal or a very low p-value or, and adjusting for multiple testing and so on and so forth. Equally important is that hypothesis that you have, right? Now, I don't mean to say that the data can't tell us a story, right? Even with low amount of data, if you do sort of pattern recognition, EDA, you might see a trend. But then the key part is that trend, does it go against the grain, right? What's the level of surprise associated with that trend? And more importantly, somewhere in the data, is there another trend or another weak trend that backs up this trend but is sufficiently orthogonal? So if you see, if you're sort of going through dozens and dozens of box plots, and if you see one box plot up, and it's like, oh, that's a signal. Or, Do you believe that? I wouldn't. But if you see something else, another association that pops up, which is weak, completely orthogonal, preferably two different assays, right? So that the noise is not correlated, right? But it sort of points to the same biology. Now that's something that I would believe that, maybe the data is telling us something that we should follow up. So it can work both ways, even in the early stages, but you have to be very careful what you're doing.
Yeah. And obviously, what you were saying earlier about sort of, having a sense of what your prior expectations are helps to guide you for knowing when you're going to let the weight of the evidence, push you in a given direction for experiment. And, also not making any major decisions in the absence of experimental validation is sort of a critical check on, whatever logical fallacies you might fall into and, interpreting the
Yeah.
data, right?
It's sort of very sort of traditional sort of Bayesian thinking, right? If you have a prior, then your data should tell you that the evidence to overcome those priors has to be very strong, right? And you have to think critically about the evidence. Obviously, if you have very weak priors or something that you've not thought about, then you have to follow the data a little bit more. And the other aspect of that is, and the other challenge of what I just described is more of the infrastructure piece is getting the data from the different vendors and from the different assays and from the different domains and connecting it to a unified data backbone, right? Or, you want to think of the unified data model or a common data model or a backbone is very critical. And every study, we have to build up that data architecture, the way you're going to integrate the data, the way you're going to connect the data. And I'm a big believer in doing that connection and then sharing the data with the clinical study teams or the translational teams so that they can go in and do their own investigations rather than us being the gatekeepers of all analysis. That's something that we've done very effectively in the past and we continue to do so is to, we are all in this together. I think our specialization is perhaps to ask some of the more complicated analysis questions like multivariate analysis, machine learning. But everybody who has access to a data dashboard that we built for them should feel enabled to go in and ask those questions or test a hypothesis quickly themselves without coming to us for a particular plot.
As we come toward the end here, I just want to end with a couple of sort of quicker questions, which is, obviously the field of data science and the field of biology, they're both moving very quickly. What are the innovations that you're most excited about in your work in the next year or 3 years?
I mean, the evolution of foundation models is something that absolutely is going to change the way we, do some research in the future, right? So we are leveraging some protein language models to sort of embed our high-dimensional data, maybe not generate new modalities per se, because we are not in that space, but, just sort of developing those encodings and doing data integration using those models is something that very exciting. Obviously, the productivity enhancement, like I mentioned, using coding tools like Copilot and others and integrating them in your system such that you are protecting your data privacy, but also then enabling gains is something that I'm very excited about. But I think the biggest thing is on the biology, right? I think using these tools, people in the research community and even in the nonprofit sector, right? They're on the frontiers of biology, just keeping up with all the papers that are coming out in the space, what we even know about the immune system, what we know about single cell biology, how tumors escape IO therapy. The scale of that, the pace of that is just amazing. And keeping up with that is one of my keen interests and something that I try to do. So technological and science, it's all going in the right direction. It's very exciting.
That's all very interesting. And then, obviously having come up in this field and, running multiple data science teams, I'm just interested in, what advice would you have for people who, want to contribute to therapeutic design and development using data-driven approaches?
I would say I look to my sort of path as well, and other folks who have been data science leaders that I worked with or I've observed. Data is ultimately, the machine learning. Everything is a tool, right? Just like doing certain experiments is a tool, right? What you do with the tools is more important than the tool itself sometimes, or at least in our sort of area of work. So I would say pick a problem that you believe in, pick a problem that interests you, right? And just go deep, understand the biology, understand whether it's clinical sciences, whether it's basic biology, whether it's something else. Go deep with that and then think about how you can leverage those tools that you have at your disposal in solving that problem. Now, that might sound scary for folks who are like, oh, what do we pick? One thing I would say is that don't worry about it. If you pick one problem for now, spend a couple of years, you might go deeper. You might say this is this is not for you, or you felt like, oh, I've done this, now I want to do something new. Options are always open. If you're very good at what you do, those doors will always be open. But just pick something that excites you, that interests you, and become an expert, become differentiated, and then you'll be just fine.
Awesome. And where can people go to learn more about you and your work?
So our company's website is ksqtx.com, and I can be found on LinkedIn.
Awesome. Well, Dipen, it's been a pleasure to have you on. Really appreciate the time and look forward to connecting down the line.
Thanks for the invitation, Ross, and thanks for the conversation as well.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.